John? John, you're on in 5. You might want to put some pants on.
Don't tell me what to do, woman. I'm head anchor around here, plus nobody can see behind the desk. I've checked.
John, you're live.
Oh, good morning, Bronx Science. This is John Burgundy with KVWN-TV. Before we get to our breaking story with field reporter Ann Sam, I'd like to remind you that KWVN-TV is sponsored by Sex Panther, cologne for men. Sex Panther is a surefire way to get the ladies. It has real bits of panther in it, and it works 100%, 60% of the time. Now to Ann.
Thank you, Ron. Right now we're in the Bronx Science Microbiology lab, and the Wingarski Hollow will be filmed right here on KVWN-TV. And to solve this mystery, I have asked expert Juliana Ross to help us unravel the secrets to the Wingarski Hollow.
Of course. Would you like me to give you 4 out of 1 Wingarski Hollow?
Of course. So what exactly is it?
Well, a Wingarski Hollow is basically a column of dirt, mud, and pond water, which illustrates the concept of microbial succession.
Microbial succession? I have never heard those 2 words before. What is it?
Well, in order for one species to survive, another must die after living for a long time and convert the territory for the others to live.
Interesting. Wow. Awesome. I can't wait to play with the dirt. So, how do you make this column?
As a scientist, you use your materials first. You have a stirring rod, some dirt, paper towels, a Wingarski column, and a beaker. My lab assistant a week ago went to the pond to get some really nice pond water. But the first step is you have to rip up the paper towels and put them in so they're 1 inch tall.
Okay, I think I'll do that.
And you also need to remove the rocks and sticks from the dirt.
Yeah, you can have fun playing with the dirt. Okay, so is this small enough?
Perfect size.
So what do you do next?
Put them in so that it covers 1 inch of the test tube.
Okay. I think I'm going to need a stirring rod for this.
Okay, take the stirring rod and pat it down so it fills the bottom.
Here, you can do that.
Okay.
Oh God, this is so much fun.
Keep ripping, Sam.
Oh, I am. Is that 1 inch yet?
When I pat it down, it'll be 1 inch.
Okay, so I'll just pop.
Perfect. Then you have to take the dirt and fill it 2/3 of the way up. You pat it down. You have to put the dirt in and fill it so it's 2/3 of the way up. Nope, you can just pour it right into the.
Okay, hold on. Let's be neat.
Okay.
Yeah, I don't think this is working that well. Wait, hold on. This thing looks like I think we should use this.
Almost definitely. It's a funnel. Here, let's pour it in.
How much do we pour in?
Until it's 2/3 of the way full.
Okay.
Poke the stirring rod so that it falls through all the way. There we go, much better.
Okay.
Keep poking.
Okay, so yeah, it's not going down.
It's okay. Scientific method always works.
Okay, if you say so.
Do I ever lie?
I don't know. Do you? Okay. That's a double touch.
Should I pour more in? Yeah, that'd be perfect.
I think you need that back.
Here you go. Okay, because this is such a failed attempt, we should show the ones that our peers made, because Wingarski columns take really long to show results anyway. So, some really good ones. are A4 and B6 because they
What
show...
are the names for? What does A4 and B6 mean?
Well, A is the morning class, which is obviously so much better, and B is the afternoon class, but you know, our color stratifications are so much better. See, as each type of microorganism grows, they make a different color in the Winogrodsky column depending on their air and nutritional needs.
The Winogrodsky column is separated into different levels: aerobic water, anaerobic water, and anaerobic sediment. As you can see, it is very complicated in the Winogrodsky column. At the top, there are cyanobacteria. They get their energy from photosynthesis. And cyano stands for blue in Greek, so that means that they are usually blue. In the anaerobic water level, there are purple non-sulfur bacteria. These bacteria are usually photoheterotrophs or photoautotrophs. This bacteria is special because they cannot use hydrogen sulfide as an electron donor for the reduction of carbon dioxide. The bacteria oxidizes sulfate. So in the next level, you can see green sulfur bacteria. This bacteria lies between the anaerobic water and the anaerobic sediment level. They are obligate anaerobes, meaning that they cannot grow in oxygen. Underneath that are the purple sulfur bacteria. It is also found in the Winogrodsky column. They are anaerobic and microaerophilic, so they require little levels of oxygen, which is perfect for them to grow in the middle of the Winogrodsky tube because the air, if there is air, it will usually be at the top. Finally, in the anaerobic sediment level, you can find Clostridium. It is a gram-positive bacteria. Clostridium is also an obligate anaerobe that produces endospore. Since they do not require oxygen to survive, the anaerobic sediment level is a perfect place for them to grow because it is at the bottom of the Winogrodsky column.
Is this normal to have some little paper at the bottom?
Yeah, because most of the paper...
Well, for this one.
Yeah, because most of the paper has been eaten away by the microorganisms, but some could still be residually left in the bottom.
Okay, so how would you further your research in this based on this simple thing?
Well, I would put it on a slide and seal it so that you can look at the specific types of microorganisms that appear in each different layer.
Oh, I also heard that like farmers do this so they know how to add like nitrogen or whatever to the soil to make it better and the crops to grow better. Is that true?
Yeah, definitely. I've seen that all the time.
Oh wow. Well, what an interesting morning of our failed attempt of the Winogrodsky column, and Juliana to help us solve the mystery of this column. Back to you, Ron.
John!
Very intriguing story, Anne. And, this is John Burgundy with KZV signing off. Good day, Bronx Science.
This was so green. What is it supposed to mean? Well, the organisms that almost require oxygen, which are facultative anaerobes, can grow in the middle of the table in the Winogrodsky column because they can have oxygen, but they could also live without oxygen. The obligate anaerobes would live at the bottom, and the obligate aerobes would live at the top.
If only humans could do that. So what about this one?
That's very similar, but there's black on the side, which indicates hydrogen sulfide production.
But this one has so much paper left. So like, what is this?
It shows that microorganisms did not digest the cellulose in the paper.
So, what does this mean?
So that means that more aerobes grew in this tube than in this tube.
Oh, okay, wow. So interesting, in this tiny little thing.
So many possibilities.
Oh, didn't you say we have to put water in it? Where did it all go?
It evaporated, and the microorganisms used it for their life processes.
Oh, okay.